In this video we're going to look at where you find living organisms and how we can count them and measure how many are there. So to start with, here's a picture of the moon landing, and I have a question for you, which is why didn't you get any polar bears on the moon? Now, hopefully you've come up with the idea that apart from being really cold and having no food, there's no air up there. So here immediately we've got a physical factor affecting where you find an organism. You won't find polar bears on the moon because there's no air for them to breathe. These physical factors affect the distribution of organisms. Distribution is just a fancy way of saying where you will find these organisms and how many of them there will be. For example, animals, there are many physical factors which will affect where you find them. The temperature of the area they're in, how available food is, how available water is, and if they're aquatic animals, how available oxygen is, because there are some organisms that can exhaust the oxygen supply in water. And finally, not so much for this leopard here, but the presence or absence of predators and also prey. Just as physical factors will affect where you find animals, they will also affect the distribution of plants. For example, the availability of light will determine where plants are found, the temperature, how many nutrients are available in the soil, how available is water, and also how available oxygen and carbon dioxide are for photosynthesis and respiration. Finally, presence of animals that might feed off them will also affect where you find plants. Now, worthy of a special mention here are carnivorous plants, plants that eat animals, for example insects. Like this here is the Venus flytrap. Now, you'll usually find them on soils with very poor nutrients, and they need to get those nutrients from elsewhere, and they'll get them from killing insects. Okay, so how do we determine how many living things there are in a certain area? We're going to look at two methods, one in more detail than the other. And the first one we're going to look at is a quadrat, which is basically just a big square. And you see how many organisms you can find within that square. For the sake of convenience, they're usually 1 meter on a side, which makes them 1 meter squared. And you can get them with some which are kind of subdivided into smaller squares to make for easier counting. Okay, so let's have a look at exactly how you can use quadrats. This is a population of little furry fuzzy ball things in a square. And if we want to count them, we use a quadrat. Now this quadrat has an area of 25 square centimeters. That will come in later on. The first thing to decide is how many samples you're going to take. Obviously, if I only took 1 or 2 samples, my results wouldn't be that good. I could happen to hit an area with a really high concentration of fuzzy ball things, or I could hit an area with very low concentration of fuzzy little ball things. By doing more than 1 sample and doing an average, you increase the reliability of your results. So for our example, I plan to do 10 samples. I've just done a little results table here. And in my first quadrat that I threw down, I found that there were 22 organisms. So I'm just going to repeat that 9 other times now. And finally, I'm going to calculate a mean based on the samples I've taken so far. So I found that my average the mean in one area of 25 centimeters squared would be 25.8 of these little fuzzy fellas. So again, just in writing in 25 square centimeters, you get 25.8 organisms. The next step is to figure out just how many of our quadrats would fit into the area we're sampling. So I'll just measure it out here. It's 30 cm by 30, which gives a total area of 900 centimeters squared. The question we need to ask is how many times would our sample area, our quadrat, and remember our quadrat had an area of 25 centimeters squared, how many times would that quadrat fit into the total area we're looking at? You have to use a bit of maths here, and you just divide the total area that you're sampling by the size of your quadrat, and it can see that I can fit my quadrat into this big square, into the total area, 36 times. Let's just check that calculation is accurate. So here I've divided up the big square into as many quadrats as I could fit in, and if I was to count them up, I would find there is in fact 36. I could fit 36 quadrats into my total sample area. So if each of our quadrats has an average of 25.8 organisms in it, and we can fit our sample into the total area 36 times, then our total estimate of the population size is 25.8, as in how many we got in an average quadrat, times how many times we could fit our quadrat in, which is 36, which gives us a total estimate of 928.8 of the little fuzzy things. So again, I decided to do a little bit of quality control and see how close to the true total I was. So each of these little paper cups contains 100 of the fuzzy little balls. And in total, there was 892. Now remember, our estimate was 928.8. So we overestimated a little bit by about 4%. Now if you look at our sample area, you'll notice that around the edges of it, there aren't as many as there are in the middle. It's not really evenly distributed. This could affect our total. In addition, the way I was counting the quadrats, I wasn't quite methodical enough about it. I didn't decide, you know, if a quadrat cut 1 in half, should I count it or not. It was a bit sloppy, really. Another way of sampling how many organisms are in an area is by using a transect. Now, a transect is very useful if you want to find how the distribution of organisms is affected as you go further away or closer to a boundary. For example, this road presents a pretty artificial line through an area. So you might notice that close to the road you've got low shrubs, and a bit further away you've got, you know, bigger trees. So if you wanted to find out how the road affected the distribution, you could use a transect. Another example of where a transect might be useful is how the distribution of different organisms is affected by their proximity to the water. So as you go further away from a river, how does the type of vegetation and animals you find will change? Finally, another example is along a coast. As you go further from the sea, the conditions change a lot, from very salty in the water and very wet to very dry and not very salty further up. So again, a linear difference. To perform a line transect, you choose somewhere to start, put a peg on the ground, tie a rope to it, and then walk off a distance, usually about 10 meters or something, till in a straight line, dragging the rope behind you. And then you just count what you've... Here's a picture of some people doing some transects along some rocks, and some bloke, I think it's a bloke, are those bubbles or hair? Anyway, he's doing a transect underwater.